Animals don't acquire nutrients as individual molecules, but instead must digest their food internally. They must extract nutrients from the food and eliminate wastes. Let's follow a meal as it travels through the alimentary canal. For simplicity, we'll divide our meal into its main macromolecules: carbohydrates, proteins, and fats. First, we'll look at the digestion of carbohydrates, which begins in the mouth. Both mechanical and chemical processing occur there. Chewing reduces the size of food particles, and the salivary glands secrete the enzyme alpha amylase. This enzyme cleaves the bonds in polysaccharides such as starch and glycogen, beginning the hydrolysis of these polymers to glucose very quickly. The salivary glands also release water and glycoproteins called mucins. Together, water and mucins form mucus, which makes the food slippery enough to be swallowed. Once food is swallowed, a wave of muscular contractions called peristalsis propels the food particles through the esophagus to the stomach. When food enters the stomach, muscular contractions churn it, mixing the stomach contents and reducing them to a uniform consistency and osmolarity. Most of the chemical processing that occurs in the stomach is the digestion of proteins, not carbohydrates. Peristalsis in the stomach walls moves small amounts of the partly digested material into the small intestine. There, secretions from the pancreas, including a form of amylase, are added to the mix. Pancreatic amylase continues the digestion of carbohydrates that began in the mouth. The cells lining the small intestine absorb the sugars that are broken down by pancreatic amylase. The epithelium of this organ is folded and covered with finger-like projections called villi. The cells that line the surface of villi have projections of their own called microvilli. The folds, villi, and microvilli of the small intestine greatly increase the surface area available for absorption. Nutrient absorption depends on the presence of an electrochemical gradient that favors the influx of sodium ions into the epithelium of the small intestine. This gradient is established by the active transport of intracellular sodium ions out of the epithelial cells, with ATP as the energy source. The movement of sodium ions out of the epithelial cells results in a low sodium ion concentration inside the cells. With this electrochemical gradient established, sodium-glucose cotransporters enable nutrients such as glucose to exit the lumen of the small intestine against their concentration gradient and enter the epithelial cells. Water follows passively by osmosis. The absorbed glucose exits the epithelial cells and enters the bloodstream, which carries it throughout the body. By the time the processed material reaches the large intestine, much of the water and virtually all of the nutrients have been absorbed. Additional water is absorbed in the large intestine, and the solid material is compacted to form fecal waste.